अस्सलाम वालेकुम डियर स्टूडेंट्स, वेलकम बैक। पिछले वीडियो में हमने देखा था मैकेनिज्म ट्रांसपायरेशन का या वाटर लॉस का मैकेनिज्म कैसे होता है। नाउ आज के वीडियो में हम बात करेंगे मैकेनिज्म ऑफ ओपनिंग एंड क्लोजिंग ऑफ स्टोमेटा। मैकेनिज्म ऑफ ओपनिंग एंड क्लोजिंग ऑफ स्टोमेटा। जो स्टोमेटा में जो स्टोमेटल पोर होता है वो कैसे ओपन या क्लोज होता है। तो जैसा कि हमें पता है कि ये ओपनिंग एंड क्लोजिंग ऑफ द स्टोमेटा जो है ये डिपेंड करती है ऑन द टर्जिडिटी ऑफ गार्ड सेल्स। टर्जिडिटी ऑफ गार्ड सेल्स, दैट मीन्स कि अगर आपको याद होगा मैंने इससे पहले भी बोला था कि ये जो गार्ड सेल्स होती हैं ये छोटी सेल्स होती हैं। यूजुअली डाइकॉट्स में किडनी शेप की होती हैं या रेमिफॉर्म शेप की होती है और मोनोकॉट्स में ये डम्बेल शेप की होती हैं। तो मैंने कहा था कि ये जो स्टोमेटल पोर है ये वॉल जो होता है, इनर वॉल्स ऑफ दीज गार्ड सेल्स दे आर थिकर एज कंपेयर टू द आउटर वॉल्स। और मैंने ये भी बोला था कि ये जो आउटर वॉल्स हैं दीज आर समवॉट इलास्टिक। तो जब भी इन सेल्स के अंदर पानी चला जाता है इनकी ये जो थिनर वॉल्स हैं ये स्ट्रेच कर जाते हैं, जिनकी वजह से सेल जो है ये टर्जिड बन जाती है। और जब ये टर्जिड हो जाती है तो ये फैल जाती हैं और ये जो स्टोमेटल पोर है वो ओपन हो जाता है। ऑन द अदर हैंड जब इसी गार्ड सेल से ये पानी बाहर जाता है तो ये गार्ड सेल जो है इट बिकम्स फ्लैसिड, ये श्रिंक हो जाती है, फ्लैसिड हो जाती है। पानी जब इससे बाहर निकलता है तो ये लॉस टर्जिडिटी करती है, जिसकी वजह से जो स्टोमेटल पोर है वो क्लोज हो जाता है। तो एक और बात ये है कि जो इम्पॉर्टेंट फिजियोलॉजिकल फीचर है इस स्टोमेटा का, हम ऐसे कह सकते हैं कि जो स्टोमेटा का इम्पॉर्टेंट फिजियोलॉजिकल फीचर, वन ऑफ द इम्पॉर्टेंट फिजियोलॉजिकल फीचर एक इम्पॉर्टेंट फिजियोलॉजिकल फीचर इस स्टोमेटा का ये होता है दैट इज रेस्पॉन्स टू द लाइट। आपने देखा होगा मोस्ट ऑफ द स्टोमेटा जो होते हैं दे ओपन ड्यूरिंग द डे टाइम, ठीक है, डे टाइम में वो ओपन होते हैं। तो वो स्टोमेटा जो दिन में ओपन होते हैं उनको हम बोलते हैं फोटो एक्टिव स्टोमेटा। और वो स्टोमेटा जो मोस्टली ओपन होते हैं इन नाइट टाइम। कुछ स्टोमेटा होते हैं जो नाइट में ओपन होते हैं, ठीक है। उन स्टोमेटा को हम बोलते हैं स्कॉटो एक्टिव स्टोमेटा, ठीक है। तो दीज आर द डिफरेंट टाइप्स ऑफ द स्टोमेटा जो फॉर एग्जांपल जो लाइट में ओपन होते हैं। इसी तरह कुछ स्टोमेटा ऐसे होते हैं जो नाइट में ओपन होते हैं। तो एक साइंटिस्ट था, उसका नाम था श्वेडनियर, ठीक है, SCHWE श्वेडनियर ने 1881 में सबसे पहले इसने बोला कि जो स्टोमेटा है द रेस्पॉन्स जो होता है या स्टोमेटल मूवमेंट का रेस्पॉन्स जो होता है इट इज ड्यू टू द चेंज इन द या इट डिपेंड्स अपॉन द ट्रिगर चेंज इन द सेल्स। That means opening and closing of the stomata. According to Schwadner, 1881, opening and closing of the stomata depends upon the changes in turgor pressure. Depends upon turgor changes. ये डिपेंड करता है टर्गर चेंजेस पे, ठीक है. एंड फ्रॉम द टाइम बहुत सारी थ्योरीज पुट फॉरवर्ड की गई इस ओपनिंग एंड क्लोजिंग ऑफ स्टोमेटा में, ठीक है. सेवरल थ्योरीज वर पुट फॉरवर्ड. बहुत सारी थ्योरीज दी गई कि ओपनिंग एंड क्लोजिंग ऑफ स्टोमेटा कैसे होता है. दैट मीन्स ये रेगुलेशन जो है, ये मैकेनिज्म ऑफ ओपनिंग एंड क्लोजिंग ऑफ द स्टोमेटा या जो स्टोमेटल रेस्पॉन्स होता है वो कैसे होता है, कैसे ये ओपन एंड क्लोज होता है. तो सबसे पहले इसमें जो थ्योरी दी गई थी वो था गार्सेल फोटोसिंथेसिस थ्योरी, ठीक है. सबसे पहली थ्योरी जो थी गार्सेल फोटोसिंथेसिस थ्योरी. दूसरी जो थी वो थी स्टार्च हाइड्रोलिसिस थ्योरी. ठीक है. जो थर्ड थी वो थी मैलेट और पोटेशियम आयन थ्योरी. So these were the chief theories which were given from time to time in order to explain the movement of the, or we can say the mechanism of opening and closing of stomata. So we will study these three theories one by one and we will try to explore what are the mechanisms behind this opening and closing of the stomata. Now, starting from this theory, that is the Garcelle photosynthesis theory. This theory was put forward by same scientist Schwadner in 1881. Schwadner in 1881, and this theory was also supported by von Mill in 1852. Actually, the idea of this theory was put forward by von Mill, but the theory was explained by this Schwadner. So, according to this theory, the guard cells, they possess the chloroplasts. Guard cells, they possess chloroplasts. So when guard cells possess chloroplasts, that means according to this theory they can perform photosynthesis. So the idea behind this theory was that during daytime sugar is synthesized. Sugar is synthesized in the guard cells. So when the sugar is synthesized in the guard cells, the sugar being osmotically active, so this sugar is osmotically active. That means when it is osmotically active, it increases the osmotic potential. Increases the osmotic potential of the cell sap in guard cells. Or we can say this sugar accumulation into the guard cells results in the decrease of the water potential of these guard cells. So decrease in water potential of the guard cells. So when the decrease in the guard cells occur, decrease in the water potential of the guard cells occur, so what happens? Water enters from the neighboring cells into the guard cells through the process of endosmosis. Through the process of endosmosis, water enters into the guard cells from neighboring cells. Whatever the neighboring cells are, either they are subsidiary cells or other cells, this water moves from higher water potential to lower water potential, because decrease in the water potential is caused by the accumulation of the sugar which occurs during the through the process of photosynthesis during the daytime. So this was when water enters these cells, these guard cells, they become turgid, guard cells become turgid, and stomatal pore is thrown open. Stomata openness. So during the daytime, it was the photosynthesis, or we can say this guard cell photosynthetic theory argues that according to this theory, during the daytime, the sugar is produced through the process of photosynthesis in the guard cells, which accumulates there. The accumulation of the sugar molecules, they being osmotically active. When the sugar is osmotically active, it increases the osmotic pressure of the cell sap in the guard cells. That's we can say that sugar accumulation in the guard cells decreases the water potential of the cells. These guard cells, when water potential of these guard cells decreases, water enters from the neighboring cells into these guard cells, and they become turgid and stomata openness. So this was the guard cell photosynthetic theory. But this was during the daytime. But in night, what happens? In night, there is no photosynthesis. No photosynthesis. But according to these scientists, according to the proponents of this theory, what happens? The sugar which is produced during the day, in the night, the sugar is converted into starch. Sugar is converted into starch. This starch, it is insoluble in water. Starch being insoluble in water. When this is insoluble in water, so we can say that it is osmotically inactive. Osmotically inactive. So when it is osmotically inactive, there is decrease in osmotic potential, decrease in OP of the cell sap. So when there is lowering of the osmotic pressure of the cells, osmotic concentration of the cells decreases, what happens? Water comes out of the cell. Water comes out into the neighboring cells from guard cells. So from these guard cells, water comes out in the process by the process of exosmosis. Through exosmosis, water comes out of these guard cells. These guard cells, they become flaccid. They become flaccid and stomata is closed. Stomata is closed. So this is the phenomenon where this conversion of sugar into starch during the night time that decreases the osmotic concentration of these guard cells. When there is a decrease in the osmotic concentration of these guard cells, so this means that increase in the water potential, or we can say increase in water potential. So when increase in the water potential of these guard cells occur, water moves out of these guard cells into the neighboring cells, and in that way guard cells, they become flaccid, they become contracted, and the stomatal pore is closed. So this was the theory given by Schwedner, and that was supported by von Michel, and in that way, so we can say this Garcelle photosynthetic theory, it gives the mechanism that due to the production of the photosynthetes during the daytime, they increase the osmotic concentration or lower the water potential of the guard cells, which results in the opening of the stomata by the entry of water into the guard cells through the process of endosmosis. And on the other hand, in the night time, the conversion of this sugar into the starch, which leads to the decrease in the water, increase in the water potential or decrease in the osmotic concentration of the guard cells, which results in exosmosis. That means water comes out of these guard cells, and that results in the flaccidity of these guard cells, and in that way stomata is closed. But there were some objections to this theory. The objections were that these guard cells possess stomata, but there's a feeble amount of photosynthesis, or we can say there is negligible amount of photosynthesis which is carried out by these guard cells. That means negligible amount of photosynthesis. Negligible amount of photosynthesis. And number second, that sugar is occasionally found. Yeah, we can say there is not much accumulation of the sugar, or we can say the sugar has not been found in the guard cells to such a concentration that they can increase the osmotic potential. Is it clear? It means we can say small amount of sugars occur in guard cells. So these were the objections raised against this theory, photosynthesis theory. Then, after that, the second theory, which was proposed regarding the mechanism of opening and closing of stomata, is that the theory of starch hydrolysis. The second chief theory regarding the opening and closing of the stomata is the starch hydrolysis theory. So, what this starch hydrolysis theory says about opening and the closing of the stomata. Now, this theory was formulated by, or we can say this was given by Lloyd in 1908. This theory was given by Lloyd in 1908, and actually this theory was formulated by Sayre. The theory was formulated by Sayre 1926. This theory was formulated by Sayre 1926, and later on this theory was modified by Stewart. This theory was modified by Stewart in 1964. 1964. So, theory was given by Lloyd, formulated by Sayre, and then this theory was modified by Sayre in 1964. So, what is the mechanism behind this theory? That we can say during the day, we can say during the daytime, there occurs the synthesis of the sugar, glucose, which is osmotically active. We know that. So, during the day, we can say that during the day, photosynthesis occurs. This photosynthesis results in the formation of sugars. Formation of sugar. Formation of sugar takes place. Now, and in the night time, the sugar is converted into starch. So we can say the synthesis of the sugars and its conversion to the starch. That means synthesis of sugar from the raw materials, photosynthetic raw materials like carbon dioxide, water, in presence of light, which we call as photosynthesis. So this sugar synthesis, and in the night time, the conversion of this sugar into starch, is the mechanism behind this theory. But how this sugar is formed during the day and how this sugar is converted to the starch into the night. So we can say that carbon dioxide, or we can say conversion of starch into sugar and sugar into the starch at night. This is the main reason behind, this is the main cause, main mechanism behind the opening and closing of this stomata. But some of the scientists like this Sayre and Scarth. Scarth was another scientist in 1926 and 1932. They argued that the formation of this, or we can say they both suggested that this conversion of starch into sugar is controlled by pH only. This is controlled by the pH of the Garcelle's. So these two scientists, Sayre and Scarth, they believe that this conversion of sugar into starch and vice versa, or starch into sugar, this is controlled by the pH of the Garcelle's. That means what is the pH of these Garcelle's when, or we can say this pH decides when there will be the conversion of sugar into starch and when there will be the opposite, that means starch into sugar conversion. Now, Stewart, 1964, he argued that this conversion of starch into sugar and sugar into starch, this is partly controlled by the enzymes and partly controlled by the pH. Enzymes plus pH. So according to the Stewart, 1964, the conversion of the sugar or the conversion of the starch into sugar, this process is accompanied by, or this process is complete by enzymes as well as by the pH. So there are two important factors behind this conversion of the sugars, that means enzyme plus pH of the cell. Now, you see, for example, पहले हम डे की बात करेंगे. During the daytime, what happens? During the daytime, जो photosynthesis होती है in presence of the light, so वहाँ पे क्या होता है? Carbon dioxide evolved in the respiration is utilized for the photosynthesis. जो the carbon dioxide which is evolved during carbon dioxide evolved during respiration is utilized in photosynthesis. So when carbon dioxide which is evolved through the process of respiration, that carbon dioxide is utilized for photosynthesis by the process of photosynthesis, that is converted into the glucose. We know that through the process of photosynthesis. So this carbon dioxide which is evolved during the respiration, that is utilized in this photosynthesis. So, and what happens, this carbon dioxide when it is utilized in the photosynthesis, it does not accumulate. It is not accumulated in guard cells. So this carbon dioxide, when it is not accumulated in the guard cells, it is used in the photosynthesis. Then what happens? The pH of the guard cells, increases to 7. So when the pH of the guard cells increases to the basic pH, that means 7, so what happens? This pH, it favors the enzymatic conversion of starch into the sugar. So उसकी वजह से क्या होता है? ये होता है, it favors the formation, this pH, that means pH 7, favors formation of glucose 1-phosphate. It favors this. That means this pH is responsible for the formation of this glucose 1-phosphate. So we can say like this. This is starch plus H3PO4 in presence of enzyme phosphorylase. It gives us glucose 6-phosphate. Yeah, it produces glucose 1-phosphate. Then this glucose 1-phosphate here the enzyme is this reaction is reversible. Here mutation occurs. Now that is phosphoglucomutase. The enzyme is phosphoglucomutase. This glucose 1,6-phosphate, it changes into glucose 6-phosphate. Now this glucose 6-phosphate by means of phosphatase enzyme, this is changed into glucose, this is changed into glucose plus H3PO4. Now you can say this starch is hydrolyzed to glucose, or we can say starch is converted into glucose, and this is during the day when there is light and carbon dioxide which is evolved during the respiration that is used in the photosynthesis. That carbon dioxide doesn't accumulate and that is changed into this glucose. So this glucose is osmotically active and it increases the osmotic concentration of the guard cells, or we can say it lowers the water potential, due to which end osmosis take place and water enters into the guard cells through end osmosis, and the turgidity increases, which throws open the stomata. Now in the night, what happens? In the night, when there is no light, that means no photosynthesis is taking place during night time. No photosynthesis is taking place, so that the carbon dioxide which is released at that time, the carbon dioxide released through different processes, that gets accumulated, that carbon dioxide is accumulated into guard cells. So we can say accumulation of carbon dioxide into these guard cells, that results in the decrease of the pH. Decrease of pH. So pH at that time is 5. This is acidic pH. So when the pH is acidic, it favors the formation of starch. So we can say that this acidic pH, it is responsible, or we can say this, for decrease in the pH, it favors the formation of this starch at the night in the stomata. So we can write the reactions like this. For example, glucose plus water, glucose plus ATP. Glucose plus ATP in presence of enzyme hexokinase. This is changed into glucose 6-phosphate plus ADP. That means this ATP, it gives the energy. Through this energy, this glucose is changed into the glucose 6-phosphate. That means the phosphate group which is added to this glucose is provided by this enzyme hexokinase. Kinases are the enzymes which add phosphate groups to their substrates. So then this glucose 6-phosphate, same enzyme phosphoglucomutase, it mutates this glucose 6-phosphate to glucose 1-phosphate. Now this glucose 1-phosphate by the action of the enzyme phosphatase is changed into starch, plus H3PO4. Starch and H3PO4. So this H3PO4, we can say this starch, as I have already told you that this starch is osmotically inactive. When this starch is osmotically inactive and carbon dioxide in the form of H3 is accumulating in, we can say, carbonic acid. This carbonic acid, this increases concentration of the carbonic acid increases, which results in the what we can say the exosmosis, or this being osmotically inactive. So water is expelled out of these guard cells through the process of exosmosis, because starch is osmotically inactive. So the osmotic concentration decrease. When osmotic concentration decreases, or we can say the water potential in turn increases, which results in the exosmosis of the cells, and in that way water comes out of these cells. Those guard cells, they become flaccid and the stomata is closed. Now also there are some objections to this theory. The objections are, first objection is that starch has never been reported in the cells of onion. Starch never reported in onion. So then how in onion stomata close and open? So this was the one objection. Second objection was the conversion of sugar to starch. Conversion of sugar to starch, it's a slow process. Slow process. But stomatal response is very quick. Stomata respond to these opening and closing responses. They are very fast, very rapid rate. So how can this slow process compete with explain that opening and closing of the stomata? And number third, glucose has never been found in the these guard cells. So these were some of the objections which were raised by several scientists today to these theories. So we can say there are several mechanisms which explain this, and the most accepted mechanism, we can say the third theory, which is active K positive ion or malate or K positive ion transport. This theory is also known as hormonal theory because of the production of abscisic acid. It's also known as hormonal theory because of production of abscisic acid. This theory was given by, actually, we can say initially the theory was given by Figino. This theory was given by actually Figino in 1959. Later on, this theory was modified by Leavitt in 1974. The theory was modified by Leavitt in 1974. So, what is the mechanism behind this theory is K positive ion. K positive ion efflux or outflux. That means entry of these K positive ions into the guard cells and expelling of these, or we can say outflux of these K ions from the guard cells is the basic mechanism behind this theory. So Let's explain this theory like this. During the daytime, what happens? Carbon dioxide concentration decreases. Carbon dioxide concentration decreases because this carbon dioxide is fixed. This carbon dioxide is fixed, and this is changed into malic acid. Malic acid into guard cells. Malic acid in guard cells. Or we can say this carbon dioxide, when fixed during the photosynthesis during the daytime, it gets changed into malic acid, which that malic acid accumulates into the guard cells. Now this malic acid is in guard cells. This malic acid dissociates into malate ions and H positive or protons. This is changed into, this is dissociated. This malic acid gets dissociated into malate ions and these H positive ions. So now when these H positive ions, they get accumulated. Now when there is so much accumulation of this these K positive ions, in order to balance it, K positive ion exchange from the subsidiary cells can take place. So K positive ions exchanged from subsidiary cells. So cell doesn't need these protons at that time. So these ions, these H positive ions, these are exchanged with the K positive ions which are present in the subsidiary cells. So when these K positive ions, they enter the guard cells, for example, we'll make like this. This is the stomata. Now these are, for example, subsidiary cells. For example, subsidiary cells have K positive ions. And inside these, there is H positive. Now these H positive ions, they are exchanged with these K positive ions. So exchange occurs like this. H comes here, K goes into the cell. When these exchanges occur, so what happens? Osmotic concentration of the guard cells increases. So the now the step is osmotic concentration of guard cells increase. When osmotic concentration of the guard cells increases, so what happens? There occurs endosmosis. When endosmosis occurs, guard cells, they become turgid and stomata is open. So this is the mechanism behind this. That means during the daytime, so we can write here the day. During the daytime, when carbon dioxide concentration decreases, because of its utilization in the photosynthesis. Now this carbon dioxide, this is changed into the malic acid in the guard cells. When this carbon dioxide is changed into the malic acid, this malic acid gets dissociated into the malate ions and H positive ions. These H positive ions are exchanged with K positive ions in the guard cells. K positive ions enter, and in the subsidiary cells H positive ions enter, and in that way exchange of these K positive and H positive ions take place. Now with the influx of these K positive ions, when these K positive ions get into this guard cell, with the help of these K positive ions, the osmotic concentration of the guard cells increases. When osmotic concentration increases, that means the cell is in need of the water, and through the process of endoosmosis, the cell absorbs water from the neighboring cells. When through the process of endosmosis, the water enters into the guard cells, these guard cells, they become turgid. They become turgid, and the stomata is thrown open. Now this process takes place during the day. Now what happens during the night? That means when during the night there is increase in the carbon dioxide concentration, because that carbon dioxide is not utilized into the photosynthesis during the night time. That means carbon dioxide concentration increased. There is increased carbon dioxide concentration during the night. Then with the help of this increased carbon dioxide concentration, abscisic acid is released. Abscisic acid is released in order to prevent the entry of K positive into the guard cells. तो इसलिए ये वहाँ पे रिलीज किया जाता है. ABA is released in order to prevent influx of K positive into guard cells. Now with the help of this, when increased carbon dioxide concentration occurs, with this increased carbon dioxide concentration, ABA, abscisic acid, is released. This abscisic acid released, it prevents the entry of K positive into the guard cells. Now after that, what happens? This K positive ion is transported back to the substrate cell. K positive back transport. Transport to the subsidiary cells. Now this K positive ion, they come back, or we can say at the night time, these K positive ions, they come back to the subsidiary cells. They are transported back because of the activity of this abscisic acid, which does not allow the entry of these K positive into the guard cells. We can say, in other words, this abscisic acid, it blocks the entry of K positive ions into the guard cells. Now what happens? Due to this K positive influx, there occurs a decrease in the pH of the guard cells. Decrease in pH of guard cells. So when decrease in the pH of the guard cells occur, due to the backflow of these, now this H positive ions, when they get more accumulated, due to which the pH decreases, pH of the guard cell decreases. When pH of the guard cell decreases, Now this pH favors the synthesis of starch. As you have in earlier mechanism seen the conversion of glucose into the starch by means of decrease of the pH. So starch is synthesized. Now this starch synthesis, it decreases osmotic potential, or we can say osmotic concentration of the guard cells decreases. Now there occurs exosmosis. There occurs exosmosis, and when exosmosis occurs, guard cells become flaccid and stomata is closed. Stomata is closed. So this is the mechanism behind this theory, that when carbon dioxide concentration increases during the night, so abscisic acid is released. This abscisic acid, this prevents the entry of K positive ions into the guard cell. So when K positive ions are expelled from the guard cell, that results in the decrease of the pH of the guard cells. So when decrease in the pH occurs, that decrease favors the formation of the starch. That starch, which is osmotically inactive, that decreases the osmotic potential, osmotic concentration of the cells, guard cells. So in that way, or we can say that increases the water potential, and increase in the water potential results into the exosmosis, and due to exosmosis, the guard cells, they become flaccid, and in that way stomata is closed. In that way stomata is closed. So these are the theories which explain these opening and closing of the stomata. Now there are so many factors which influence this opening and closing of the stomata. Several factors are there, like for example, or we can say these are the factors influencing stomatal movement. So yeah, stomatal activity. So which are the factors which influence this stomatal activity? So first of all, the main factor is light. So you can say most of the stomata which open during the day, as I have already told you, the photoactive stomata, those open in the light. Now there are two types of light, for example, blue and red. These both influence the activity of the stomata, but this blue is most active in opening. Or we can say when there is a blue light, it induces the opening of the stomata. Although red is also there, which can induce the stomatal opening, but not as much as blue. So we can say blue has a dominant role in opening of the stomata. Now the second factor is temperature. Usually if we will see 25 to 35 degrees centigrade temperature, it favors the opening of the stomata. But if the temperature is too much, we can say that is too much low, that is towards the freezing, that causes the closure of the stomata. That means closure of stomata occurs at freezing temperatures. Or even too much temperature, like 45 degrees Celsius, it also causes closure of the stomata. And the third reason is humidity. If the environment is humid, much humid, it favors opening. Or we can say higher humidity results in the opening of the stomata, and lower humidity, that means dry air, it causes the closure of the stomata. Dry air causes the closure of the stomata. Now, fourth one is water availability. If water is in a good amount, that favors the opening of the stomata, and if there is water scarcity, we can say if the water content in the soil is too low, it causes the closure of the stomata. Or we can say lower water content, it causes the closure of stomata. However, higher water concentration, higher water availability, stomata are open. After that, one more factor is carbon dioxide concentration, CO2 concentration. Lower CO2 concentration favors opening, while as higher CO2 content, it causes the closure of the stomata. At higher levels of carbon dioxide, stomata remain closed, while as in lower concentrations of the carbon dioxide, stomata are thrown open. And in that, then there is pH, one more factor, and you have already seen if the pH of the guard cells increases, that favors the pH, favors higher pH. Higher pH favors opening, because at higher pH there occurs sugar formation, glucose formation by the activity of the enzymes, and if the pH is lower, it favors starch formation, that leads to closing of the stomata. So we can say at higher levels or higher pH, the stomata are open, and at lower pH, the stomata are closed. And then there are some hormones, growth hormones. If we will take the case of cytokinins, these cytokinins, they favor the opening. Cytokinins favor the opening of the stomata. However, abscisic acid, it favors the closure of the stomata, or it causes the closure of stomata. Because you have seen, according to that latest Lewis theory, it prevents the K positive influx into the guard cells, due to which the starch pH decreases, then that causes the synthesis of the starch, and which leads to the flaccidity of these guard cells, and the stomata are closed. Now, one more factor is, that means mineral ions. Some of the mineral ions like K positive, as you have already seen its role, that this regulates the stomatal movement by throwing them open. That means K positive favors opening of the stomata. And in the same way, there are several other minerals like phosphorus, nitrogen, calcium, which also play a role in opening and closing of stomata. So In that way, we have completed this topic, that is the mechanism behind the opening and closing of stomata. Thank you.